thank you all so much for signing on and checking out my talk this morning. I'm very excited to share some of my PhD work from my time at the University of Georgia, which focused on the temporal dynamics of phytoplankton. Oh, it's not shared?
No, it's good. I just needed to click on a different thing. Sorry, carry on.
Yeah, I can see your screen, Sean. It looks good.
Okay, good. I'm just trying to minimize the control panel, sorry. All right, let's try this again. Okay, so yes, my talk's going to be on the temporal dynamics of phytoplankton mortality in the Skidaway. So my research at UGA focused on marine protists, which are single-celled microbes that greatly influence the structure and function of marine ecosystems. Now, marine protists are incredibly diverse in terms of their size, shape, and function. They can gain their energy in a number of ways, such as autotrophy, which means they use sunlight and photosynthesis to fix carbon, heterotrophy, which means they consume other cells, and mixotrophy, which is a combination of both. Now, phytoplankton, which are autotrophic protists, are responsible for nearly 50% of global primary production and oxygen production. And as a result, phytoplankton fuel life and mediate carbon transfer within marine food webs. Now, due to their ecological importance, it is critical to understand the fate of phytoplankton populations, which relies on the balance between their growth and mortality. So currently, our knowledge of phytoplankton growth exceeds that of mortality, with reliable drivers such as sunlight, nutrients, and temperature shown to impact phytoplankton growth. My research has focused more on the other side of this, so phytoplankton mortality, namely three main sources: microzooplankton grazing, viral lysis, and parasite lysis. And though my research has only focused on these main three, I do want to mention that there are many other forms of phytoplankton mortality that I will not cover, such as grazing by larger zooplankton, programmed cell death, and physical sinking. But of these main three, the most dominant form of phytoplankton mortality is grazing by microzooplankton, which are 20 to 200 micron heterotrophic protists that consume 60 to 70% of daily primary production. Now, microzooplankton can ingest a wide range in prey size, from cyanobacteria all the way up to chain-forming diatoms. Here's an example on the bottom of a dinoflagellate grazer before and after ingesting a much larger centric diatom. And then also, microzooplankton can often grow at rates that are on par with their phytoplankton prey, which allows them to rapidly exploit and control phytoplankton blooms. Though not as dominant as microzooplankton grazing, viral lysis is responsible for 10 to 20% of daily primary production loss. Viral infection is host-specific and density-dependent, and viruses can also terminate phytoplankton blooms, transfer genes from host to host, and alter host biodiversity and function. And now on the bottom, we have some examples of viruses that are attached to a phytoplankton host cell. And unlike these other two forms of mortality, we do not know much about parasite lysis, though we do know that these protist parasites are widespread and have been shown to infect a range of different plankton groups. Now, parasite infection is thought to be driven by host density and encounter, with highest infection happening during a phytoplankton bloom. Some of the stages of infection are now shown in these bottom panels. In a dinoflagellate host Akashiwu sanguinea, panel A shows a healthy Akashiwu cell. In panel B, the arrow is pointing to a parasite spore, which is called a dinospore, that has infected the host cell. And then finally, panel C shows late stage of infection, where the parasite has grown to almost fill up the entire host cell. Still, it has remained hard to characterize parasite-host dynamics, as most parasite species are either unknown or have yet to be brought into lab cultures. All right, so these different forms of phytoplankton mortality can each have a major top-down impact on phytoplankton populations, influencing their abundance, diversity, and composition. Furthermore, these different loss factors can have vastly different impacts on carbon cycling within marine food webs. For instance, microzooplankton grazing, which is at the center of this food web in blue, typically leads to the transfer of organic carbon up the food web to higher trophic levels, whereas viral and parasite lysis, as shown in red and green, lead to the direct input of host particulate and dissolved organic matter, which can then be reused by heterotrophic bacteria. So ultimately, it is important to understand these different forms of phytoplankton mortality to accurately predict shifts in plankton populations, biogeochemical cycling, and related ecosystem health. Now, phytoplankton mortality, especially microzooplankton grazing, has been well documented throughout the world's oceans. So each red dot on this map represents a dilution experiment where grazing rates have been measured. And so you can begin to understand the true spatial coverage of these rates. However, compared to extensive spatial sampling, there have only been a handful of experiments that have measured grazing rates over time at the same spot. And even fewer experiments have considered how virus or parasite dynamics may change over time. Well, why should we care how phytoplankton mortality changes over time? Well, for one, this temporal variability in mortality is realistic for many coastal regions, such as estuaries, where phytoplankton populations and environmental factors also vary over time. Moreover, these temporal measurements may allow us to identify the different abiotic and/or biotic drivers of phytoplankton mortality, which in turn may inform how mortality changes in a future climate. All right, so this brings us to my three main PhD chapters. So chapter 1 focused on the short-term variability in plankton mortality rates at same-day high versus low tide. Chapter 2 on seasonal variability in plankton mortality rates on weekly scales. And then chapter 3 looked at temporal variability in parasite-host populations. So before we dive into chapter 1, I just want to give you guys some brief background on the sampling site and methods that I used. So I performed all of my water sampling in the Skidaway River Estuary at the UGA's coastal campus, which is on Skidaway Island, and that is shown by the red dot on this map. Now, the estuary itself is quite shallow, 4 to 6 meters, well mixed, and has a 2 to 3 meter tide range every 6 hours. And while previous sampling in the estuary has revealed both temporal shifts in environmental factors and phytoplankton, plankton mortality dynamics remain poorly understood here. So at the center of my work was a year-long time series where I gathered surface water samples from the estuary using a Niskin bottle, which you can see here in the top two images. And basically what I was doing is gathering surface water each week from February 2017 through 2018 and using this water to perform dilution experiments to measure microzooplankton grazing, phytoplankton growth, and viral lysis rates. And I was also collecting water for 18S amplicon sequencing. During a period of 8 weeks, from June 22nd through August 17, I ramped up sampling and performed a dilution experiment at same-day high versus low tides to measure short-term shifts in phytoplankton rates. This will be covered in chapter 1. For chapter 2, I performed a dilution experiment each week at high tide to measure seasonal shifts in phytoplankton rates. And lastly, for chapter 3, I switched gears to explore temporal shifts in parasite host populations using weekly 18S amplicon surveys. All right, so some more methods. So to measure phytoplankton mortality rates, I used the dilution method, which was pioneered by Landry and Hassett in 1982 and has since become the predominant method for estimating in situ and simultaneous rates of phytoplankton growth and microzooplankton grazing. So let's briefly walk through this method. So we first take a whole seawater sample from the estuary that has both grazers and prey. We then dilute this 100% whole seawater sample with filtered seawater from the same source that does not have any grazers or prey. So in theory, the more dilute fraction, in this case 20% whole seawater, will have less encounter between grazers and prey, and in turn, lower microzooplankton grazing. We then take these triplicate 20 and 100% whole seawater bottles. We incubate these bottles for 24 hours in a nearby flow-through tank. At T0 and T24, we then measure bulk chlorophyll and the concentration of different phytoplankton groups using a guava flow cytometer, shown here on the bottom left. The flow cytometer measures three major phytoplankton groups: the cyanobacteria Syndiniales, as well as picoeukaryotes and nanoeukaryotes. And this is based on plots of their fluorescence versus side scatter, which is just a proxy for particle size. And then we can calculate the daily change in either bulk chlorophyll or plankton concentration, assuming exponential growth, which is referred to as lowercase k. The change in chlorophyll, or lowercase k, is plotted versus the fraction of whole seawater, and we fit a straight line through these points. And then after all this work, we can find the in situ phytoplankton growth rate in the absence of any grazing, which is taken to be the y-intercept, whereas the microzooplankton grazing rate is the slope of this line. Now, I do want to mention that there are two primary assumptions with the dilution method. The first being that grazing is linear to the dilution, which means there's less grazing happening at the more dilute prey levels, which in our case was 20% whole seawater. The second assumption is that phytoplankton growth itself is not impacted by the dilution of whole seawater. To make sure that this held true, we did add nitrates and phosphate to our bottles, and also had a 100% whole seawater treatment without nutrients added. Lastly, we set grazing rate equal to 0 if the slope of this line was not significantly different from 0. We also used some common modifications. So we added a treatment of 20% whole seawater that was diluted with ultrafiltered water to remove both viruses and grazers. The slope of this new line then became the total mortality rate, and the viral mortality rate was inferred as total minus grazing rate. So normally the dilution method involves setting up many more fractions of whole seawater, though we only use 2 points, 20% and 100%. And recent work has shown that rates from a 2-point method are comparable to a full gradient and reliable to nonlinear grazing. And as you have seen, this method can be quite lengthy. So using fewer bottles and fewer sampling points allowed us to sample more often. Okay, and then finally for methods, so on top of the dilution experiments, I also took surface water samples each week to measure some other properties, such as temperature, salinity, dissolved oxygen, bulk chlorophyll, nutrients, particulate organic carbon, nitrogen, solar radiation, and flow cytometry. I also ran flow cam samples each week to measure the biomass of the larger 20 to 200 micron plankton. The flow cam was able to capture thousands of plankton pictures per 10 mil sample, some of which are shown here on the bottom right. And I do want to point out that nutrients and POC-PON were not measured at low tide, and so were excluded from chapter 1. So this brings us to my first chapter, which focused on the short-term variability in plankton mortality rates at same-day high versus low tides. And findings from this journal, or findings from this chapter, have been published in the Journal of Limnology and Oceanography. So let's jump right in and first look at rates based on bulk chlorophyll. So on the top graph, we have grazing rates on the y-axis. Bottom graph, we have phytoplankton growth rates. Sampling days on the x-axis, which will be true for most of the figures in chapter 1. And for all figures in chapter 1, high tide is going to be in dark blue, low tide is going to be in orange. So based on bulk chlorophyll, we see that grazing rates were significantly higher at same day low tide compared to high tide on 5 out of 8 days, while there was no clear trends in phytoplankton growth rates between high versus low tides. There was also no significant viral lysis measured based on bulk chlorophyll. All right, so let's look at the same types of plots, but now for pico-eukaryotes on the left and Syndiniales-Protist on the right side. And as with chlorophyll, the top 2 panels are showing you grazing rates, the bottom 2 are showing you growth rates. So for both of these groups, we found that grazing rates were again higher at low tide compared to high tide, this time for both groups on 7 out of 8 days. While again, there was no trends in phytoplankton growth rates between high versus low tides. Viral lysis rates, as shown here by the faded bars, were measured for both pico and for syn, though with no real trends between high versus low tides. And I also want to point out that we also found no trend between tides for nano-eukaryote grazing rates. So that graph is not shown. So for 3 different groups, for chlorophyll, Syndiniales-Protist, and pico-eukaryotes, we see that microzooplankton grazing rates were higher at same day low tide compared to high tide. So what is driving these patterns in grazing rates that we see at high and low tides? So we found that grazing rates were poorly explained by most abiotic factors, with linear regressions having low R-square values ranging from 0.03 to 0.24. We did find a weak positive relationship between bulk chlorophyll and microzooplankton grazing rates, which you can see here, with an R-square value of 0.36. However, grazing has been shown to vary based on phytoplankton prey species, and so bulk chlorophyll is thought to be a poor proxy of microzooplankton grazing. And then, like grazing, growth rates are poorly explained by most or by all abiotic factors, with low R-square values less than or equal to 0.2. So these changes in microzooplankton grazing rates may have instead been driven by observed shifts in plankton biomass. So here we have a plot of biomass on the y-axis in units of micrograms of carbon per liter for the major plankton groups measured via flow cam and flow cytometry. Again, sampling days on the x-axis. And now for each day, the left bar is going to be high tide, the right bar is going to be low tide. So a couple of things to point out here. First, we see that the biomass of the mixotrophic dinoflagellate, Akashiwu sanguinea, which is in orange, was higher at low tide on all sampling days, especially on June 28th, July 6th, and August 3rd. Now, Akashiwu has been shown to rapidly ingest less than 20 micron phytoplankton prey, which may have explained the higher grazing rates that we measured for pico-sized phytoplankton at low tide. We also found that diatom biomass, shown in green, was higher at high tide from August 3rd through 17, with these diatom populations mainly composed of the chain-forming genus Skeletonema. And this is also important because prey type, so prey size or shape, can influence grazing rates, which may have been the case in some of our experiments where these larger chain-forming diatoms were present at high tide, but not at same-day low tide. So to further quantify shifts in plankton composition, we performed an NMDS. So each point on this NMDS plot represents the log transformed biomass that was measured via flow cam. Points are labeled on the NMDS by sampling day. And to try and quantify shifts in plankton composition between high versus low tides, we ran an ANOSIM, which computes an R value, with R values closer to 1, meaning that tides were more different from each other. So while we did find a low R value of 0.22, plankton composition was significantly different between high versus low tides, and you can see that low tide samples are less clustered overall compared to high tide samples. So therefore, this variability that we observed in grazing rates at high versus low tides was likely driven by measurable differences in both plankton biomass and composition. And one thing that I do want to point out is that while we did not quantify water mixing, such vertical and horizontal mixing is likely going to impact which phytoplankton species or types are going to be present at each tide. All right, so this short-term variability in microzooplankton grazing rates can have a major impact on how we look at phytoplankton populations. So now on this graph on the y-axis, we have net accumulation rates based on bulk chlorophyll, which is just growth minus grazing rates. And again, sampling days on the x-axis. So from this plot, we see that the accumulation of plankton biomass is very different at same-day high versus low tides, often being higher and positive at high tide. So on average, over this 8-week period, these accumulation rates were 0.46 per day at high tide, negative 0.37 per day at low tide, and 0.05 per day if we combine both high and low tide. Well, let's say that we went back and only sampled at one single tide, and then used that average rate value to estimate temporal shifts in phytoplankton biomass. So if we were to plug these average rate values into an exponential growth equation with a starting chlorophyll value of 5 micrograms per liter, we would see that at high tide, chlorophyll would increase exponentially. At low tide, chlorophyll would decrease slightly. And if we were to combine all values from high and low tide, chlorophyll would be fairly stable. So with just this simple exercise, we can see that that rates that you measure, even those taken 6 hours from each other, can have a major impact on how we estimate plankton biomass and related carbon cycling. And thus, it may be important to consider such tidal variability for accurate daily rates, especially within these highly dynamic regions like estuaries. All right, so let's move on from looking at short-term rates in Chapter 1 to now look at how rates change on weekly scales for Chapter 2, which again were measured at only one tide, so at only high tide from February 2017 through 2018. And findings from Chapter 2 have been published in Frontiers in Marine Science. Okay, so let's first look at rates based on bulk chlorophyll. So on the y-axis, the growth rates are going to be the closed circles, grazing rates are the open circles. Again, on the x-axis, we have sampling day, and now seasons are shown below that. So overall, for bulk chlorophyll, we see that phytoplankton rates varied between seasons, with higher growth and grazing rates in summer versus fall and winter. Though measured for on all sampling days, viral lysis rates were only observed on 2 out of the 41 days, as you can see here by the red stars. Now on the bottom, we again have a plot of accumulation rates based on bulk chlorophyll, which again is just growth minus grazing. We see that these accumulation rates were higher in summer and fall compared to winter and spring. And often in springtime, phytoplankton or microzooplankton grazing was higher than phytoplankton growth, which meant that more phytoplankton biomass may have been consumed at this time. All right, so let's look at the same types of graphs, but now for pico-eukaryotes on the left in orange and nano-eukaryotes on the right in purple. So for both of these groups, pico and nano, we see the same seasonal trends in growth and grazing that match that of bulk chlorophyll, with higher values in summer relative to fall and winter. As with chlorophyll, viral lysis rates were only observed on 3 to 4 days for each of these groups, as you can see by the red stars. Again, on the bottom plots, we have accumulation rates based on both of these groups. And we see the same trends that match that of bulk chlorophyll, with higher values in summer and fall relative to winter and spring. I do want to mention that grazing rates on Syndiniales were only significant on 5 out of 41 days at high tide. So for chapter 2, for 3 different phytoplankton groups, bulk chlorophyll, pico-eukaryotes, and nano-eukaryotes, we see the same seasonal patterns in both growth and grazing rates, where you have higher values in summer compared to fall and winter. So what is driving these rate patterns? So out of all the environmental factors that we tested for, phytoplankton growth correlated most strongly with surface water temperature, which is what you see here, where you have temperature on the x-axis, phytoplankton growth is on the y-axis. For the 3 major groups, chlorophyll, pico, nano, note that I'm using the same color scheme. So for all 3 of these phytoplankton groups, we see that linear regressions were significant, with R-square values ranging from 0.46 to 0.56. And in general, you can see that phytoplankton growth increases with higher temps. Now, this is not a major shock, as temperature has been shown to be an important driver of phytoplankton growth, though we may also expect that temperature is an important driver of microzooplankton grazing. But we found that grazing rates were poorly explained by surface water temperature for all 3 groups, with low R-square values ranging from 0.11 to 0.27. So in our case, grazing was poorly explained by surface water temperature or any other environmental factor. However, as we saw in chapter 1, microzooplankton grazing rates can also be influenced by biotic drivers, such as plankton biomass and composition. Okay, so here's a plot again of plankton biomass in units of micrograms of carbon per liter for the major plankton groups that were measured via FlowCam and flow cytometry. Sampling data is again on the x-axis, and seasons are shown below. So I don't have time to go over trends for each of these groups, but want to point out an important shift that happens in phytoplankton biomass. So in winter and spring, we see that biomass was lower, less than 400 micrograms of carbon per liter, and dominated by less than 20 micron combined phytoplankton, so Syn, Pico, Nano, which is shown in blue. Whereas in late summer and fall, biomass was higher and dominated by chain-forming diatoms such as Guinardia and Chitoceras, which are shown in green. So let's zoom in and just look at the biomass of those 2 groups. So diatoms versus less than 20 micron phytoplankton. And that's what you see here in the top graph. Note that biomass is now on a log scale. And on the bottom graph, we again have accumulation rates based on bulk chlorophyll. So we see that this shift to diatom biomass that happens in late summer and fall does coincide with an increase in phytoplankton accumulation rates, where phytoplankton growth exceeds microzooplankton grazing. So diatoms have been shown to avoid grazing, either by forming these large spines or chains or by releasing toxins, which may have explained why we found microzooplankton grazing rates to be so low at this time. But overall, what this shows us is that shifts in phytoplankton composition, so in this case diatoms versus less than 20 micron phytoplankton, can influence grazing rates and may have masked the impact of any single abiotic factor. So as with chapter 1, we performed an NMDS to quantify seasonal shifts in plankton composition. I know this plot looks kind of crazy, so let me walk you through it. So each point on this NMDS plot, again, represents the log transformed biomass that was measured via FlowCam. Points are labeled by sampling month and day. And to look at both week to week as well as seasonal shifts, we used a natural color gradient for our samples, where red samples were in winter, purple in spring, blue in summer, and green in fall. So as with chapter 1, we wanted to quantify shifts in composition between seasons now. So we again ran an ANOSIM, this time with ANOSIM R values closer to 1, meaning that seasons were more different from each other. So while we did see some overlap happening, plankton composition was significantly different between all seasons with an R value of 0.48, and was most different between fall and winter and spring and fall with R values of 0.64 and 0.61. We also ran a SIMPER test to try and find out which of the FlowCam groups may be most responsible for these shifts, and found that shifts between fall and winter and spring and fall were most driven by the chain-forming diatom Chaetoceros. So overall, these shifts in phytoplankton populations were likely important in driving seasonal microzooplankton grazing rates. And the real take-home for chapter 2, the real take-home for chapter 2 is that while phytoplankton growth may be driven by these overarching factors, such as surface water temperature, this may not hold true for microzooplankton grazing, which are also driven by shifts in the density and composition of their phytoplankton prey. Okay, so just to briefly recap so far for chapters 1 and 2, we found that temporal shifts in grazing rates were poorly explained by any single abiotic factor and were instead likely driven by observed shifts in both grazer prey composition and biomass. Viral lysis rates were seldom observed for all phytoplankton groups, and one explanation of this may have been that our incubation time of 24 hours was too short to capture the full viral infection cycle, which has been shown at times to extend past 24 hours. And then this work from chapters 1 and 2 reinforces the idea that grazing rates are not static. And so it'll be very important to incorporate this temporal variability in grazing to allow for more accurate monitoring of plankton and predictions of their population dynamics within coastal food web models. So to this point, we have looked at microzooplankton grazing rates and viral lysis rates, though unlike these two sources of mortality, parasite lysis rates are not yet captured by the dilution experiment. So to get at parasite-host dynamics, we used weekly 18S amplicon surveys. Okay, and this brings us to my last PhD chapter, which focused on the temporal variability in parasite-host populations. And this chapter is currently in review at the M-Sphere Journal. Okay, so because we are shifting gears, let's go over some brief background on parasites. So the most dominant group of protist parasites are Syndiniales, which are marine alveolates, and they often represent the most abundant taxa within global 18S surveys. Now, Syndiniales have a rapid infection cycle of 2 to 3 days, and they release hundreds of spores called dinospores following the infection of a host cell. So here's a simplified diagram of their infection cycle using stained samples of a host and a parasite cell. So let's briefly walk through this process. So the parasite starts off as a free-living dinospore that can infect a new host cell. Once inside, the parasite grows to form what is called a trophont, which is in red and green, inside of the host cell, which is in blue. The trophont will grow until it ruptures through the host cell and releases what is called the vermiform. And then finally, the vermiform will fracture and release hundreds of new dinospores that can infect new phytoplankton hosts. Now, Syndiniales have been shown to infect many different plankton groups, especially dinoflagellates. Here's some examples of dinoflagellate species that have been infected by Syndiniales. In all panels, the arrow is pointing to the parasite trophont, which has infected the host cell. And I just want to point out this last panel, panel 7, which shows a chain of Alexandrium cells. And you can clearly see that the inner 2 cells are healthy and much smaller compared to the outer 2, which have been infected by Syndiniales. Now, although Syndiniales are expected to be an important source of plankton mortality, there remain many questions on their population dynamics. For instance, how do Syndiniales change over time? What drives these temporal changes? Who do they prefer to infect, and how specific are these interactions? So let's briefly go over the workflow for chapter 3, which is highlighted by this simple flow chart. So on 33 out of the 41 sampling days, I filtered triplicate surface water samples and extracted DNA. I then used primers to target the V4 region of the 18S rRNA gene, and then sent samples for amplicon sequencing on a MySeq at the UGA bioinformatics core. Now, I'm not going to go into great detail on the bioinformatic processing, but I'm happy to answer any questions that you have later. So I used data2 in R to infer amplicon sequence variants down to 1 to 2 base pairs. ASVs were annotated using the PR2 database, and we ended up with around 9,800 ASVs from all of our samples, and around 7,400 ASVs after filtering out metazoans and ASVs that were only found once. Lastly, we converted ASVs to relative abundance, and then protist composition and diversity were explored using a suite of packages in R, such as phyloseq, vegan, and ggplot2. Okay, so let's first look at the relative abundance on the y-axis of the major class level groups. You will note that the bar plots are separated and not stacked to more easily visualize temporal shifts for each group. Classes that were less than 5% on any sampling day were grouped into other, which is top left in dark gray. And on the x-axis, sampling day is grouped by month, which will be true for most of the figures in chapter 3. So of all these major class level groups, only Syndiniales, shown on the bottom left in purple, exhibited strong temporal variability, making up 7 to 28% of the relative abundance from June to October, compared to less than 6% in winter and spring months. Now, Syndiniales themselves were composed of 3 main groups, which I'll refer to from now on as dino groups 1, 2, and 3. And as with microzooplankton grazing rates from chapters 1 and 2, we wanted to test out which abiotic factors may be driving shifts in Syndiniales. For this, we used a PLS, or partial least squares, which is a technique used when you have many co-varying factors, which we had, and seeks to find the factor or factors that best explain a single variable, which in this case was the relative abundance of Syndiniales. So of all the environmental factors that we tested for, temperature was shown to be an important driver of Syndiniales. Given its importance as a driver, we chose to use surface water temperature to better look at temporal shifts in protist diversity and composition. So let's first look at Shannon diversity for all 18S ASVs, and that's shown here on the y-axis. Again, sampling days on the x-axis and grouped by month. We used a temperature-based color gradient for our samples where lighter colors, so greens and yellows, represent warmer temps, and darker colors, blues and purples, colder temps. So for all 18S ASVs, we see that Shannon diversity was fairly stable, dropping only in April and in January. We also performed an NMDS to look at shifts in the total 18S composition. The NMDS has the same color gradient, but now shows triplicate samples on each day. So for the NMDS, we see that samples taken during warmer months from June to October formed a tighter cluster compared to colder months. All right, let's look at the same 2 plots, but now for Syndiniales ASVs only. So for Shannon diversity, we see the same trends. It's fairly stable, dropping only in January. And for the NMDS, we see that Syndiniales composition forms an even tighter cluster in these warmer months from June to October. So to verify these trends, we also ran a CCA using environmental factors and Syndiniales composition, though the environmental factors only explain 19% of the variance in Syndiniales composition. And as we saw in the first 2 chapters with grazing rates, this may show us that temporal shifts in Syndiniales may be more driven by the biological interactions that they have with other protist groups. So to explore these biotic interactions, we constructed a co-occurrence network using a program called CONET. And very briefly, CONET uses a combination of pairwise measures such as Pearson, Spearman, and Bray-Curtis to find significant interactions or edges between ASVs or nodes. And basically, these interactions are based on patterns in the relative abundance profiles for each ASV. So here's a quick and simple example of how CONET works. We start with two nodes or ASVs that are connected to each other by an edge, which signifies an interaction between these ASVs. As you add in more and more ASVs, the program is going to find more edges that are between these ASVs. For our network, we only used ASVs that were found in greater than 50% of our samples. Circular networks were visualized in a program called Cytoscape and were filtered to include only edges found between Syndiniales and other protist groups. Based on the sign of the interaction, edges were either positive, which I will show as dark blue from now on, or negative, which will be in orange. We focused on positive interactions between Syndiniales and other protist groups that may infer parasite infection, though in general, these positive edges could imply mutualism or grazing or simply an overlapping niche between ASVs. And to that end, it's very important to understand that while these networks can predict interactions, they cannot confirm them. Okay, so we found that Syndiniales were well represented in our network, making up 20% of all edges. So here's our lovely circular network that has been filtered to only show significant edges with Syndiniales. You will see that ASVs are grouped on the outside by class. Classes that only had one ASV were grouped into other, which is in dark gray. And Syndiniales ASVs are at the very bottom, shown in purple. And one more thing to point out is that in this case, thicker lines mean that there were more interactions found between Syndiniales and the other protist groups. So right away, we see that Syndiniales had positive interactions in blue with a range of different protist groups, such as Dinophyceae, which are dinoflagellates, Bacillariophyta, which are diatoms, and Spirotrichia, which are ciliates. Now, although this may not look quite as cool, this bar plot offers a more manageable breakdown of this network, showing the total number of positive and negative edges between Syndiniales on the y-axis and the major groups on the x-axis. So a few things to point out here. First, we see that positive interactions were most common between Syndiniales and Dinophyceae, which is in line with frequent microscopic observations of infection within dinoflagellates. Positive interactions were also common in Spirotrichia, which are also known to be observed hosts of Syndiniales. And then lastly, CONET lets you input abiotic factors, though in our network, abiotic factors, which is shown in light gray, had far fewer edges with Syndiniales compared to the biotic interactions with other protist groups. All right, so let's zoom in now and just look at parasite-host dynamics between Syndiniales and their most common host, Dinophyceae. So now we are looking at a presence versus absence matrix of all the significant edges that were found between these two groups. You will note that Dinophyceae ASVs are on the top, with species IDs shown when possible, and Syndiniales ASVs are on the left and labeled by group, so dino groups 1, 2, or 3. So from this matrix, we see that positive interactions were found between Syndiniales and a range of dinoflagellate species, as shown by the blue stars, such as Akashiwu, Gymnodinium, and Heterocapsa, all of which have been involved in confirmed parasite infections through prior fieldwork. We found only a single one-to-one positive interaction between Syndiniales ASV 43 and Dinophyceae ASV 20, which was Akashiwu sanguinea. Here's a look at the temporal profiles of these two ASVs. So in purple, you have Syndiniales ASV 43, and in green you have Dinophyceae ASV20. So we can see that this ASV pairing was strong over time with a Spearman R value greater than 0.8, and both ASVs peaked from June to September. More often, a single Syndiniales or Dinophyceae ASV had multiple interactions with more than one ASV from the other group. So for example, here's the same type of plot, which now shows a single Dinophyceae ASV28 in green, which was heterocapsa, shown here, and two Syndiniales ASVs in purple, 27 and ASV 77. We see that both of these different ASV pairings were also strong over time with a Spearman R value greater than 0.7. So what this shows us is that Syndiniales and Dinophyceae interactions may be flexible, and that Syndiniales may exhibit a more generalist method of host infection. Still, these interactions and infection dynamics need to be tested further, for instance, through more targeted lab experiments using a range of parasite and host taxa. So just to recap for chapter 3, we found that Syndiniales were temporally variable with highest relative abundance from June to October, where it was up to 28%. Though clustered based on surface water temperature, Syndiniales composition was poorly explained by abiotic factors. We found Syndiniales to be well represented in our network, making up 20% of all edges, having positive interactions with confirmed dinoflagellate species, as well as potential hosts such as diatoms, and often with greater than 2 hosts per parasite ASV. And while parasite lysis is thought to be an important source of phytoplankton mortality, it is rarely incorporated within marine food web models. Therefore, gathering this baseline information on Syndiniales and their hosts in both the lab and field offers a great first step in assessing their role in phytoplankton mortality. Just to wrap everything up from my PhD work, so we found that phytoplankton mortality, grazing rates, and parasite dynamics varied over time in the estuary. Grazing rates and parasite populations were poorly explained by any single environmental factor, and instead were more likely impacted by observed shifts in grazer-prey composition, biomass, and biotic interactions between taxa. And as we saw with high versus low tide rates, when you sample can matter. And so it is important that we keep making these temporal measurements of phytoplankton mortality to better understand shifts in plankton and carbon cycling. And then very, very quickly, I just want to touch on the next steps for my new role as a postdoc here at AOML, which is going to build on the field sampling and amplicon sequencing work for my PhD. So here we again have a simplified workflow of processing of marine eDNA, with eDNA representing all DNA found in a water sample from single-celled bacteria all the way up to skin cells that are released by fish and marine mammals. Now, more and more science labs are sampling for marine eDNA, which is awesome, but it has been hard to compare findings between labs, as they often will employ their own slight modifications to this general workflow. So one of the goals of the omics program here at AOML is to streamline this eDNA workflow from eDNA sampling and extraction all the way up to bioinformatic and data processing. So one of my first projects here at AOML will be to try and optimize these first 2 steps, so eDNA sampling and extraction, focusing on how the extraction of eDNA may impact these more downstream steps, which includes key measurements of species diversity and composition in bacterial and all the way up to fish populations. And so with that, just want to thank past and present members of the Harvey Lab, which was at UGA, is now at UNH, my committee members at UGA and funding sources there, and happy to take any questions that you may have. Thank you.
Thanks, Sean. That was great. That's a lot of work I can see, and it's nice how it came together and lots of different trends that you could explain. So does anyone have any questions? I think we have a small enough group that we can probably just, speak out loud, or if you want to type, that's okay too.
Yeah.
I'm sorry for being late. I got the wrong link that was on the calendar. I'm sorry, but I thought that was a great presentation.
Oh, thank you. Appreciate it.
I was wondering how you would, so you had some great trends that you saw, but for the most part, they were within a single year. And I'm sure like
Yep.
there was a tremendous amount of work and you were limited by the time of your PhD, et cetera. But I guess I'm wondering how much you think the trends that you saw were just dependent on the particular year that you were there, or that you sampled, and how you might expect it to what more you could learn if you had multiple years. And I guess what I'm thinking about the eDNA work that we're doing, how you might use some of different technologies or kind of long-term sampling going forward for the eDNA work, and for using maybe some of the technologies like that Ian and Nate have come up with, for example.
Yeah, so to get at your first question, I think it definitely would have varied based on year. So there was a paper that did kind of similar work. They didn't measure rates, but they looked at phytoplankton concentrations and different factors in the estuary. I think it was in 2012 when they sampled through the whole year. And that year had very high rainfall. They actually did 2 years, 2012 and 2013. One of the years had extremely high rainfall and high drainage into the Skidaway River Estuary. And so they saw vastly different, chlorophyll and different plankton concentrations and species composition from year to year, just based on like water drainage. So I think our year in 2017 was a dry year. It didn't rain. We didn't have like too many storm surges or anything like that. So I think in that location, rainfall could definitely play a huge role on, phytoplankton rates. And then to get at your second question, I wish I had a SASE. That'd be awesome if I could go back and use something like that for this work. I think, if you do want to do a temporal study, then you definitely, it takes time to do these dilution experiments and this field work. But to look at eDNA over time on like a weekly scale or even tidal scale, you could employ an instrument like the SASE, which Ian and Nate are working on, and that's one way that you wouldn't have to be out in the field for the whole day. So that would be awesome.
Yeah, no, absolutely. I think your talk gives a really nice justification or a use case of where something like that would be really, really useful.
Yeah.
So Chris has a question. Chris, do you want to ask your question, or I guess I can read it?
Yeah. Yeah, I see it.
So it says, would you like to further address next steps, especially the need for optimization of sample processing for different types of environmental matrices, sediment, seagrass, water, or different types of water masses, varying turbidity or environmental inhibitors?
Yeah, that's a lot. That's a loaded question. Yeah, I think that there's definitely a need for processing of different types of, water in environments. I haven't worked in sediment or seagrass, but I think definitely within an estuary too, those are critical components. One of the things that was an issue is that you have a lot of grazing that happens on the bottom of the estuary by bivalves, and so trying to do this sampling at different depths in the water column, maybe closer to the bottom sediment, would be awesome. But I think that's definitely warranted, and you would see something different there. Different water masses. Yeah, I mean, we didn't measure turbidity. I think that's something that is hard to quantify, especially in the estuary that I was working in. And then environmental inhibitors. Yeah, I'm not quite sure what you mean by that one. Environmental.
Yeah, I mean, I guess there could be different like with sediments, for example, or soils in the terrestrial realm, there might be certain compounds that would inhibit like DNA extraction. I think he's asking specifically about
for the DNA extraction. Yeah.
DNA. I mean, the rates is definitely another challenge in different environments. Probably that assay that you talked about would be more for pelagic kind of situation with encounter rates and whatnot. But yeah, looks like he's talking about DNA extraction and things that inhibit the enzymes and or the polymerases and things like that. So fortunately, we don't. Well, in a lot of the stuff we're doing at AOML, we don't have those kind of more troubling materials. But yeah, so I don't know if you want
and then for those,
on that or
then for like, for those different, for sediment, seagrass, obviously you'd have to change your extraction protocols, and so it'd be a bit different, but yeah.
All right.
Questions?
Any other questions before we wrap it up? Okay, well, Sean, you're hearing virtual applause now. Thanks to Sean for the great talk, and
great job, Sean.
Thanks, guys.
We will, it's all right with you, we'll make it, I'll keep this recording and maybe we can share it with folks that weren't able to join.
Yeah, awesome.
Okay. All right, well, see everybody next time.
Thank you, guys.
See you guys.